to CS Squared News. Today we will be talking about plant cell structure, photosynthesis, cellular respiration, and protein synthesis.
We hope you enjoy.
Today we'll be talking about the plant cell, not the animal cell.
I will hear from Sarah Stanley about the structure of the plant cell.
Thank you, Caira. Plant cells are eukaryotic cells that have a cell wall, which is the outermost layer of the cell, and it protects the cell from infections. The plant cell contains several organelles which are held in the cytoplasm of the cell. Some of these organelles include the Golgi apparatus, mitochondria, chloroplast, smooth and rough yard, and the nucleus. The chloroplast is where photosynthesis takes place. The chloroplast contains thylakoids, where the light reactions take place. Sunlight and H2O goes into the thylakoids and produces O2. The Calvin cycle takes in CO2 and produces sugars, which is the point of photosynthesis. These two processes take place in the stroma of the chloroplast. The mitochondria is where cellular respiration takes place. Glucose goes through glycolysis, the Krebs cycle, the electron transfer rate to produce ATP. Now on to Cindy Douglas with cellular respiration.
Thank you, Sarah. Cellular respiration is the process of turning our food into energy. This part of the cellular respiration takes place in the mitochondria, and this part takes place in the cytosol. Glycolysis turns glucose into pyruvate, which then gets oxidized by acetyl CoA. It then goes to the citric acid cycle, where electrons get carried by NADH and FADH2. Then it goes through oxidative phosphorylation and produces ATP. The first step of cellular respiration is glycolysis, which is the breakdown of glucose to pyruvate. For each molecule of glucose, 2 ATP are added and 2 NADH are produced. 4 molecules of ATP are produced by substrate-level phosphorylation, and finally, 2 pyruvate are formed. The pyruvate from glycolysis combines with coenzyme A to produce acetyl CoA. 1 NADH molecule and 1 CO2 molecule are produced from this. During the citric acid cycle, 2 carbon molecules are lost and CO2 is produced, which is what we exhale. FADH, NADH, and GTP are formed. A result of the citric acid cycle is a 4-carbon acceptor molecule that is regenerated for each step. The molecules produced from the citric acid cycle go through oxidative phosphorylation. This is the process of producing ATP from NADH and FADH. The electron transport chain passes along electrons from NADH and FADH2. Throughout the chain, electrons give up energy used to phosphorylate ADP to ATP. NADH provides electrons that have enough energy to generate 3 ATP and FADH2, which generates 2 ATP. The last electron acceptor on the electron transport chain is oxygen. The half O2 accepts the 2 electrons and together with 2 H pluses forms water. Now to Caira Lefdahl for photosynthesis.
Photosynthesis is the conversion of light energy into chemical energy through the sunlight. There are 2 stages of photosynthesis, and they are known as light reactions and the Calvin cycle. Light-dependent reactions occur in the thylakoid, and light-independent reactions occur in the stroma. Photosynthesis is a process by capturing free energy in sunlight and storing that energy in chemical bonds, especially glucose. This equation of photosynthesis is the reverse of the equation for cellular respiration, except that the energy required by photosynthesis comes from light. The processes are interconnected. Energy stored in chemical bonds by photosynthesis is then extracted from those bonds by respiration to make ATP. The energy in ATP is then used to do cellular work through metabolic processes. The process uses plants take carbon dioxide and some water and sunlight and turn it into sugars or carbohydrates. Plants are able to harness photons from the sunlight and then turn them into sugars that we can then eat and we can use for energy.
And now for the process of photosynthesis. Water goes through the stomata, which are the pores of the leaf, goes into the thylakoid, and releases ADP and NADPH. Oxygen is released, CO2, ATP, and NADP go into the Calvin cycle and releases to make sugar.
And now on to Sarah Stanley for protein synthesis.
Protein synthesis is a process where amino acids are arranged into proteins, the ribosomal RNA, transfer RNA, messenger RNA, and various enzymes. Proteins are synthesized on ribosomes and help catalyze reactions of cellular respiration and photosynthesis. The process starts out with DNA and then uses 2 processes known as transcription and translation. Transcription takes the information encoded in DNA and encodes it into mRNA, which heads out of the cell's nucleus and into cytoplasm. During translation, the mRNA works with the ribosome and tRNA to synthesize a protein. After the protein is synthesized, it goes to ribosomes, rough yard, transfer vesicles, Golgi apparatus, or whatever is needed. If it is sent to the Golgi, then it is modified and packed into secure droplets.
I'm Caira.
I'm Sydney.
And I'm Sarah.
Thank you for joining us on this week's broadcast of CS Squared News.
Catch you next time.
Watch us next week for our video on sexual reproduction.
Are you hungry? I could use a light snack.